Good morning, everybody, and thanks for the opportunity to speak on this topic. As was mentioned, I'm a metabolic physician at the Children's Hospital of Philadelphia. So what that means is that I take care of a lot of kids with rare diseases that essentially disrupt biochemical pathways. So in my clinical practice and in my research program, we use biomarkers all the time. We love biomarkers. We love measuring the amounts of different chemicals or molecular substrates in our patients to try to understand what's going on. So today, I will use some of those examples, both from our clinical practice, but also as we think about research development, to sort of highlight what biomarkers can and cannot do, and then also think about how you apply those biomarkers in a clinical trial setting. So here's an outline for what I'm going to speak about today. Really starting with this first topic of how can biomarkers be helpful, and it was set up in such a beautiful way from our first talk. Some of this will be repeated, but hopefully we'll be building on to think about the specific roles of biomarkers. So starting there, what are the potential functions of biomarkers? This has actually been fairly well defined by the FDA and the NIH, and it was alluded to already, this idea of the best resource, which essentially defines the biomarkers, endpoints, and other tools that you can think about in a clinical trial. And I want to speak through some of these different tools or some of these different ways that a biomarker can be used and helpful for your trial. And I'm actually going to refer a lot to this review article that I have highlighted here, which I had no part in writing, but I think is a wonderful resource if you're trying to learn more about biomarkers. It focuses here on neurodegenerative disorders, but the concepts described here I think can be applied to lots of different diagnoses. So here on this somewhat complicated table, we have all the different ways or contexts of use in which you can use a biomarker. And we're going to go through and think about what these definitions actually mean. And to do that, we're going to use a real-world example of one of my favorite biomarkers, which is something called phenylalanine. So I take care of a lot of kids that have a disorder called PKU, or phenylketonuria. This is a disease where you can't convert phenylalanine to tyrosine. And what happens is that phenylalanine builds up. It's just an amino acid that builds up and is toxic to the brain. So phenylalanine has been used as a biomarker for both clinical practice and really for studies for decades. And so I think it actually checks off a lot of these boxes that we speak about in terms of context of use. It's been used so many different ways. So we're going to talk through just as an example so you can understand all the different things a biomarker can do. So first off, one thing a biomarker can be useful for is diagnosis. And actually, every baby born in the United States has this biomarker checked through newborn screening. And this is actually a picture of my youngest getting his newborn screen. But Phe levels are detected off of a newborn screening dried blood spot. And those babies that have high phenylalanine levels are referred into a center like mine and usually, typically, diagnosed with PKU. So this is a biomarker that confirms the presence of a disease. So phenylalanine is very helpful for that. But it's not only used for that. It's also used as a prognostic biomarker. So what a prognostic biomarker does is to identify the likelihood of a clinical event or to help you figure out how severe the course might be for a patient. So in the case of phenylalanine, when we see patients in clinic, we then get a confirmatory level. And if that level is less than 360, we think that this is probably something called benign hyperphenylalanemia and they don't need further treatment. But if it's higher than 360, they fall in the category of classic PKU. So essentially, this biomarker at day of life 7 or 5 or whenever I meet these babies can tell me how severe I expect the clinical course to be for a patient. But it can do even more than that. It also can show a pharmacodynamic response. So what we mean by this is that the biomarker can change in response to some sort of intervention or medical product. So here's just some data from our preclinical work that we do on PKU, where we have mice that have very high phenylalanine levels, shown in orange and green, that have PKU, essentially. And we give them a base editing therapy. It's a therapy we're trying to develop in the lab. And you can see the mice that got the treatment in orange, very quickly, that phenylalanine level that was very high, starting around 2000, normalizes. So essentially, we're using the phenylalanine to measure a response to a potential therapy. So that's a pharmacodynamic response biomarker. And then finally, another thing that we use phenylalanine for in PKU is for monitoring. So a monitoring biomarker is one that can be measured over and over and over again to figure out the status of a disease or a medical condition or the effect of a medical product. And so our patients with PKU do finger stick or heel stick blood draws every couple of weeks to make sure that their Phe levels are staying in a normal range. So as you can see, phenylalanine plays a lot of different roles when you start thinking about biomarkers. And if you sort of categorize all the different things a biomarker like phenylalanine can do, you can see that it can both quantify elements of the disease target and elements of a biologic response to something, whether that be an intervention, a drug, a diet, whatever it is that you're trying to assess. So in terms of the disease target, it can help you identify risk and diagnosis and prognosis. And then when you think about a biologic response, is the drug that you're trying to develop actually engaging the target that you want? Is it modifying the disease? There are other biomarkers that we haven't talked about here that we use all the time in clinical trials, things like safety biomarkers to make sure that what we're giving to a patient is indeed well tolerated and safe. Okay, so those are some of the potential functions of biomarkers. But what I think a lot of people in this audience, especially a lot of my advocacy colleagues, are thinking about is how can they be used in the approval of a new drug? And we've had some nice discussions about this already this morning. But I think it's important to remember that the drug development process has a very long timeline. But biomarkers can guide every step of this path. So let's say very beginning in the lab, and you're trying to identify a potential compound or drug or gene therapy approach that might be helpful for your disease. You can use them in screening assays in cells in a dish, for example, to see if your drug or your compound is changing some sort of meaningful outcome, in this case, a biomarker. But as you move through your non-clinical studies, things like animal studies, biomarkers are often used to figure out if your drug is acting the way we should or the way you anticipate it would. You then move into phase 1 trials where safety biomarkers become incredibly important, but also target engagement. Is the drug actually hitting the pathway or the thing of interest that you're trying to correct? You then move to phase 2 and 3, where you start really using biomarkers to measure efficacy and safety at the same time. And as was alluded to and discussed beautifully, biomarkers are incredibly important in the regulatory review process, which comes after those first clinical trials. And then, as I've alluded to, as part of clinical care. So we know that biomarkers, like I said, are used for diagnosis and monitoring and safety monitoring. But also, as a physician, I look at biomarkers all the time to make sure that whatever drug I'm using in my patients are actually making a difference. So it's used at every step along the way. Okay. Can a biomarker primary endpoint support the approval of a product? And we've talked about this a little bit, but a biomarker can be an example of a surrogate endpoint. And the FDA defines this very carefully. They say surrogate endpoints can be used or are used instead of clinical outcomes in some clinical trials. But there's caveats to this. They're used when the clinical outcomes might take a very long time to study. So for example, I study some disorders where it takes 10 years for the disease to progress, and no industry sponsor, no academic investigator can afford, in all honesty, to run a 10-year trial. So it's important to think about surrogate endpoints. Or they can be used in cases where the clinical benefit of improving the surrogate endpoints, such as controlling blood pressure, is well understood. And finally, and I want to highlight this point, you need some sort of clinical trials or clinical data to show that the surrogate endpoints can be relied upon to predict or correlate with a clinical benefit, right? You want to make sure your biomarker is measuring something that's actually beneficial to a patient. And that can be the tricky point sometimes. So let's go back to our example of phenylalanine. Are there data out there? Do we know if phenylalanine levels actually are good surrogate endpoints? Can they be relied upon to predict clinical benefit? And there's been so much work in this field. It's pretty amazing if you look in the literature trying to make this association. I picked one study, and there's no way you can read the small text on this, but I show you that this is just a clinical trial where they did a battery. Each of those lines is a different neuropsychological test. So in PKU, people can have problems with executive functioning or neuropsychiatric concerns. So they essentially did a series of 12 or more tests on adults with PKU, and then correlated how many tests the patients had problems with. So how many tests did they essentially fail, and how did that correlate with phenylalanine levels? So the x-axis here is showing how many tests the patients had, an individual had difficulties with, And the y-axis is showing how many tests the patients had difficulties with. And the x-axis is showing you the phenylalanine levels. And you can see patients with higher phenylalanine levels did worse on more tests. So this is just one piece of evidence that try to link this biomarker measured in blood to something that's clinically relevant or have a clinical benefit for a patient. And so this surrogate endpoint has been used actually for the approval of, at least at this point, 2 different PKU drugs. The first, a cofactor drug, Kuvan or cephalosporin, back in 2007, and then more recently, pegvaliase in May 2018. And so this has actually been a clinical trial endpoint that has supported approval of drugs that patients take today. If you're curious what other surrogate endpoints have been acceptable to the FDA, there's a really nice table that's kept up to date on the FDA website that essentially shows you all the different surrogate endpoints that have been the basis for a drug approval or licensure. And it's really interesting to scroll through and look at your favorite diseases or biomarkers of choice. So I highly suggest you take a look. Okay. So what role do patient communities play in biomarker selection and validation? And I think this is really important to consider, especially in the world of rare and ultra-rare diseases where I practice. So, like I said, the FDA said clinical trials are needed to show that surrogate endpoints can be relied upon to predict or correlate with an actual clinical benefit. But I think it's important to remember, how do we define clinical benefit? And who should decide what is clinical benefit, right? Should it be myself as a physician who maybe sees a patient in my office for an hour, every other month? Or should it be the families that are actually taking care of these amazing kids on a regular basis? And I will say I'm a little biased, but I think it should be a combination in a group decision that we work together in order to build that decision. But how do you do that? And we've talked about this a little bit already when we talk about patient-centered outcome measures. And I think many in this room are familiar with this concept, and it was introduced beautifully already. But this idea that you start actually asking the families or the patients, if they're able, what would be a meaningful clinical benefit? How do you define clinical benefit in something that's actually going to help a patient? And then as you think about validating your biomarkers, making that connection between the biomarker and the clinical benefit, you can incorporate some of these patient-centered outcome measures. It's important to capture what's actually going to be helpful to the families. So I highlight that because I think it's a team effort to think about biomarker development. Okay. In the last part of my talk and my remaining time, I'm going to be doing sort of a case study of developing a biomarker for an ultra rare disease that I think about a lot in my practice, multiple sulfatase deficiency. And I think you might see some overlaps when we think about some of the biomarkers with what you're going to hear about from some of our subsequent speakers thinking about MPS disorders. But hopefully you'll see this is the process, and my thought process, at least, and our team's thought process of how we tried to do this moving forward. So where do you start, especially in an ultra-rare disease? Well, the first step is building a team. And the team that we have built, or I have been honored to be a part of, was actually built by Alan Finglas and Amber Olson, who are 2 patient advocates. I think many of you in your room probably know who they are. And as Alan always says, you got to get the right people in the boat to start the discussion. And over the past 8 years, he's been working really hard to try to get the right people in the boat. And so this is just an old picture when we're all much younger at a conference in Dublin in 2017, where we met to discuss essentially how do you go from a point in time where we had essentially no therapeutic entities under development, no biomarkers, not a real understanding of the natural history of the disease, to first-in-human clinical trials. And he recruited a number of colleagues of mine into the boat in order to start thinking about how do you validate a biomarker. And we knew at that point that this was going to be essential if we were going to move to clinical trials. So he recruited 2 of my good friends and colleagues, Lars Schatala from Germany and Laura Dang from CHOP, and a lot of this data will be a collective effort of all 3 of us. Okay, so what is multiple sulfatase deficiency? What is the ultra-rare disease we're talking about? Essentially, it's a deficiency of an enzyme that activates all cellular sulfatases. These are enzymes that are really important for breaking down things in your cells. When you lose that activating enzyme, you end up essentially having broken enzymes or decreased activity of 16 different sulfatases, many of which are associated with single enzyme disorders. So essentially things you might have heard of that are a little more common, like MPS disorders, like Sanfilippo, a couple different forms of Sanfilippo syndrome, like MPS II, or metachromatic leukodystrophy. So you can see biochemically, this is a complex disease, and you actually have the potential for lots of different biomarkers. There's so many enzymes that are not working well in this disease. Lots of stuff is building up that might be a good biomarker. Now, when you lose the activity of all these enzymes, unfortunately, you end up with a very severe progressive multisystemic disorder that's super complex. And so you need to start thinking about what actually would be clinically meaningful given the incredible burden of disease that our patients experience. So we had a hypothesis going into this that the biomarkers reflecting the most impaired sulfatases, essentially those enzymes that were not working the most, will be the most effective diagnostic and severity biomarkers for MSD. And hopefully down the road, a prognostic biomarker, or the ability to see whether or not it's changing in response to a therapy. But starting out, we really had no idea. So before we could tackle a biomarker, we had to figure out what would be a meaningful clinical benefit in MSD. And as was already said so beautifully this morning, when you have an ultra-rare disease, you have to make the most of the data from every single patient. And so we tried this for a long time, using every retrospective data, chart, patients, any information we could get about any patient with MSD. And these are essentially two different studies that we published looking at retrospective data. And we have an ongoing prospective natural history data study that we're working on right now. But from these original studies, we found that while lots of organ systems can be involved, we think the most clinical benefit might come from fixing the neurological symptoms of this disease. And that's because, to show over on the far side in A, almost all patients have neurological symptoms. So this is a uniform problem across the whole population. It's very burdensome, and it is very severe. Now, the rest of the somatic features, we call things like the heart, the lungs, the bones, were much more variable. So we thought, in all honesty, they might not be the best things to link a biomarker to. So when we hone in on the neurologic function, we wanted to be able to figure out how to actually quantify a change in neurologic function, which is not an easy thing to do, especially in neurodevelopmental and neurodegenerative conditions. But we were lucky that there's a scale for a related disorder, again, borrowing from other rare diseases, for metachromatic leukodystrophy, that could capture the loss of gross motor function. So all this is showing you essentially is that at 0, that means a patient can walk typically, and then as they lose skills and sort of fall down that slope at 6, they no longer have much neurologic function. And we found that this could actually capture the progression of disease. So as the clinician and the investigator, we thought, okay, linking a biomarker to this could be very useful. But what if the families actually want? So in parallel, some of my colleagues at CHOP were thinking about what caregiver priorities would be in this population. And this is a little hard to read, but I'll show you the top gray tranche of essentially how many people, what the most relevant topic were, all related to comfort and neurologic comfort, right? So the ability, I see Sarah shaking her head, our patient advocate partner. But yes, essentially making sure that patients' neurologic comfort was a priority. And so while we had been focused on motor, they were focused appropriately so on comfort. And we thought that maybe it would be important if we're going to pick a neurological biomarker to make sure it links with both of these goals. Okay, so once we sort of had a target in mind, the next thought we had to figure out was, are there actually abnormal biomarkers in multiple sulfatase deficiency, which meant actually looking. And so we are lucky in that we had patient samples from our natural history study and the MSD Foundation-led biobank, and a lot of friends who helped us along the way to basically first figure out, classify patients into more severe or less severe patients, and then do a variety of biochemical assays to figure out if we could find a biomarker that would segregate with severe or attenuated disease, and then maybe in the future, a response to a therapy. And I'm not going to go through all the enzymatic data that Mike Gelb and others did for us, which has been amazing, but I will show you that essentially we could figure out different enzyme activities that could differentiate the most severe patients from controls and the mild patients from controls, which is really important when you start thinking about diagnoses. But not all enzyme behaved equally. And the enzymes that actually affected the brain, so things like the enzyme responsible for Sanfilippo syndrome or the enzyme responsible for metachromatic leukodystrophy, did much better in terms of differentiating severity groups, again suggesting that the brain is important. So once we looked at the enzyme levels, we moved on to thinking about what builds up when those enzymes are broken. And this is important because those are actually more reliable, going to the point before, in terms of being able to do over and over again without differences in the laboratory. And we looked to see if those GAGs, those things building up, could be diagnostic and severity biomarkers. And this is just showing you that I think we'll speak about this more in the Sanfilippo discussion, but in a disorder like MSD, lots of different things build up. So you have all this stuff that can't be broken down, these GAGs, and you can see that in each individual sulfatase disorder, like MPS II or Sanfilippo A or Sanfilippo D, a unique species build up. But in MSD, everything builds up. But what we found is that essentially in MSD, the stuff that's building up in the brain, the neurologic biomarkers, were by far the best thing at differentiating cases from controls and severity groups. So we think they might be the best diagnostic and prognostic biomarkers. But again, that's just a biomarker. You have to actually link it to something that has a clinical benefit or clinically meaningful. And so to do that, we then started laying over those regression maps that I showed, essentially the loss of motor skills. And we found that patients that had higher markers, higher GAG accumulation of those key biomarkers shown in black, tend to progress earlier than patients that had that more attenuated forms, or the lower levels of those biomarkers progressed it later. So we think that this is the first sort of step toward developing a severity biomarker and hopefully a disease response biomarker. So what's next? How do we know if these actually will capture clinical benefit? We are very lucky that we're moving towards 2 first-in-human clinical trials in this disease, one through of AAV gene replacement by Steve Gray's group and Rachel Bailey, and then another of drug repurposing in the EU. And what's great is these are both efforts, the Bespoke Gene Therapy Consortium, which supports the AAV program, and Remedy for All, which is an EU-based drug repurposing program that is trying to develop a playbook or a map for how other rare diseases can do this. So we hope that the biomarkers that we move forward and actually sort of unify across the 2 trials will show rare disease communities the way to move this forward. So in conclusion, I think it's important when you think about a biomarker as either a surrogate endpoint or as a supporting feature of a clinical trial, you have to make sure that your biomarker, first of all, captures the right target, could be the right target organ, the right target cell type, whatever it is of interest to you, that the biomarker helps you select the right drug and the right dose. Finally, also to help identify the right patients, going to the point earlier that different patients in the disease progress or different patients at different steps along the disease progression might need different biomarkers, and then help you design the right trial. And that's really the goal of what we are hoping to do with biomarkers. And I have lots of folks, if this goes forward, that I'd like to thank. Most importantly, our patient advocacy groups. It's an amazing community in the MSD world, and our funders and our collaborators. And I'm happy to take questions.
Thank you very much. I think we have time for maybe 1, maybe 2 questions. I think we have one right over here.
Yeah. Hey, Becca, great talk. So I'm curious, when you showed the correlation between the GAG levels and the attenuated versus the progressing group, it seemed to distinguish them well, but I'm guessing that probably clinically you knew that anyway at diagnosis. So I guess my question is, are they distinguishing the rate of change of those different patients in the attenuated group, which I'm guessing would be a bit more clinically relevant to say, okay, you're going to progress at this rate or that rate?
Yeah, that's a great question. We are doing those serial collections now. I will say at diagnosis, you can't actually always predict who's going to be severe versus attenuated. Most of our diagnoses now are coming through exome sequencing in babies that might have a little bit of developmental delay or feeding difficulties, and we don't know which rate they're going to progress on. But it is very important, as you mentioned, to do serial analyses of those biomarkers to figure out if they actually can help guide sort of a disease stage measurement or something along those lines. But the other thing that's really important is, do they change in response to a therapy? And right now, like I said, we haven't been able to do this in patients yet, but in animal models, it does look like they're a great biomarker of a pharmacodynamic response with some of the preclinical programs we have. Thanks.
Another question.
Thank you for your talk. So kind of a general question. When you have a biomarker with proven clinical benefit, but this biomarker is metabolized, and you have, so your biomarker has metabolites, and those metabolites may have actually opposite effect of your biomarker. So what is the balance here to make?
Let me repeat the question. I'm having a hard time hearing you. But
So when you have a biomarker with clinical benefit, and this biomarker end up having metabolites, and your metabolites have actually opposite effect of your biomarker. So what balance you would have to make?
I think that's a really good question. I think you have to balance essentially over time how those metabolites are changing and whether or not they're actually either mitigating your clinical benefit that you're seeing or influencing the clinical benefit. But again, that only comes when you have the luxury of doing those studies and trying to make that link between whichever biomarker, whether it be a metabolite level or a primary biomarker, and the clinically meaningful change that you're hoping to see.
Thank you.
Yeah.
Thank you very much. Thank you for the questions, and thank you, Dr. Ernst Nicholas.